So basically what I want to do today is share with you some results from our analysis of single cell RNA sequencing of small cell lung cancer. To give you some context, small cell lung cancer is a very lethal disease. It's the worst type of lung cancer you can have, and the reason is that it has a shorter doubling time than non-small cell cancers. Um, it metastasizes earlier, and so what that means for patients is that usually by the time they're diagnosed, the cancer's in the extensive stage. It's spread through the lymph nodes and even to the brain, and it's too late to do much. The 10-year survival rate for this disease is only 3.5%, and the standard of care hasn't changed substantially in decades. And a big reason for that is cellular heterogeneity. Uh, in this context, what I mean by that is that the tumor or tumors undergoes some sort of treatment. Some of the tumor cells die, but the tumors that live proliferate, regrow, and metastasize and continue to cause havoc throughout the body. So what we'd like to understand is how those tumors that survive the treatment differ from those tumor cells that are killed by the treatment. And so single cell RNA sequencing gives us a tool to do that. And so what's usually done with this type of data is that the raw data measures all the genes and all the cells. You usually have thousands or millions of cells, and you have, depending on the model organism, you have 20,000, 40,000 genes. What's usually done is highly varying genes are selected first, and those are the genes that vary the most, and we make the assumption that those are the most important. Then principal component analysis is done on that set of highly varying genes. And then finally, a nonlinear dimensionality reduction is done to visualize. And what's typically done is that the clustering and the cell type identification and most of the analysis is done after principal component analysis or after t-SNE. But there's a problem with that. And to see why that is, consider the example of the 3D Earth being represented on a two-dimensional map. If you want to represent the whole surface of the Earth, you either have to blow up some land masses like Greenland, so that's much bigger than it actually is, or appears much bigger than it is, or you have to tear the map so that one part of Greenland is way on the other side of the map than the other. And so when you reduce the dimensionality of data, you always introduce distortion. And so one of the things that we've done in the DEEDS lab is we've developed a tool to quantify this distortion. And so what you have here, if you can see in this top frame, that the dimensionality reduction for this S curve has left the neighborhood structure intact. The red cell or the red points are close to the red points, the orange points are close to the orange points, et cetera. But with the sphere, the neighborhoods are more broken up. And so there's more distortion. And so what we do is, I don't have time to explain how this measure works, but we have a quantitative way for evaluating this distortion, and we can apply it to data. And when we do that for single cell RNA sequencing data, what we found is that this workflow introduces huge amounts of distortion into the data, where as much as 97% of the nearest neighbors are different than they are in the raw data, which is a problem because most of the analysis techniques, the algorithms, use a nearest neighbor search of some type. So if we want to analyze our data, what we've started doing in our lab is we do our clustering and our cell type identification on the raw data, and then we do dimensionality reduction just for visualization, just to look at what we get. And so we have 8 cancer cell lines. These are immortalized cell lines for small cell lung cancer, and the sequencing was done by our colleagues at Vanderbilt. And when we apply our workflow to these samples, what we see is that there's a lot of heterogeneity in each one of these samples. And so our mission after getting this data was we wanted to figure out what exactly this heterogeneity corresponded to. The first thing we did was we did a manifold learning technique to make sure that what we were seeing wasn't just an artifact of the clustering, and that there actually was a difference in structure. And so we applied this technique where we take PCA at differing embedding dimensions and measure the distortion. And as you can see, one of our clusters has a much lower curve than when compared to a random subsample of the same size. And so what that means is that there's a difference in complexity between these clusters. And so it's already known in the literature that small cell lung cancer is falls into generally 4 types, or at least that's the understanding right now, is that there's at least 4 subtypes. And so the first thing we wanted to know, in each one of these cell lines, are we seeing these clusters? Do they correspond to the known subtypes? And when we did our gene analysis, we found that there wasn't a significant difference in the marker genes that are supposed to indicate these subtypes in our clustering. So the heterogeneity that we're seeing here is heterogeneity other than the difference in subtypes that was previously known about. And we found that there are other genes that we found in an unsupervised way that can indicate which one of these clusters a given cell might belong to. And so biologically, we wanted to figure out what was going on here. So one of the first things we wanted to see is we wanted to make sure that this wasn't just because the cells were in a dish. And so we measured the gene expression for epithelial mesenchymal transition marker genes. And basically what we found was that there wasn't a statistically significant difference between clusters for these genes. So whatever we're observing here, it's not EMT transition. We also, on a hunch, we looked at chromatin remodeling genes, and we found that for these genes, these sets of genes, that there actually was a statistical significant difference between the genes for these clusters, leading us to believe that there was a different chromatin remodeling program going on in these clusters. And that led us to our next step, which was we used a tool from Stanford called Cytotrace. And this tool is trained on stem cells, and it gives a score between 0 and 1, with 1 being the most stem-like and 0 being the least stem-like. And we found that in our cluster that differed the most, we found that the probability of this algorithm to find that these were stem cells was much higher than in the others. And so our working hypothesis is that these cells are some type of proliferative cell that is more proliferative than cancer cells on average, but we still have a lot of work to do. In our lab, we develop a lot of tools for single cell RNA sequencing. So we're working on new quality control techniques, because one key question is how much of what we're seeing in this raw data is noise and how much is actual biological variation. We're working on new clustering techniques that can take some of our pre-existing biological knowledge into account for the clustering. And we also, we're working on new manifold learning techniques, because we want to better characterize the system of gene regularization, and hopefully we can translate that into something that will be useful for patients. And we also want our collaborators to do these experiments. We basically want to treat cells with chemotherapeutic agents and sequence the survivors, and the same with radiation, so that we can see if this heterogeneity changes, or if it's the same after treatment, and hopefully understand something that can be therapeutically useful. And so I'd like to acknowledge my colleagues in the DEEDS lab, especially Tim Hamilton and Samuel Aragones, because they've worked really hard on this project, and they've done a lot. And so with that, I'd like to have any questions.